Hi everyone, I'm Jen Harland and I will be chairing this final session. So we have our keynote now by both Joe Porter and Andrew Want, talking about marine life in the wrecks and blue carbon habitats.
And thank you very much for the kind invitation to speak at this very special commemoration event. I work up at the ICIT Heriot campus here in Stromness. So my talk this afternoon links very closely to the exhibitions that are ongoing in the Stromness Museum. And earlier in the year I was working very closely with Jeanette and Katie to put together the marine life part of the exhibitions. And so some of the stuff I'll be saying in the next few minutes will relate to some of the artifacts in that exhibition. So if you haven't seen it already and you've got time, please do go and have a look around. So I'd like to start by saying that how Scapa Flow is now from an environmental point of view is quite different to how it was a few years ago. And I want to take you back to the Pentland Firth Tidal Atlas. And there were surveys done by Commander Cortland H. Simpson, RN. And these surveys were very basic surveys, but gave us an idea of the soundings and also of tidal flows coming through during that period. The flow at that time was quite different because obviously we didn't have the block ships and we didn't have the Churchill Barriers. So we can see that we're getting water flowing through quite strongly in areas where it doesn't flow anymore. And particularly relating to Mark's talk earlier on, this area between Lambholm and Glimpsholm, where obviously it's very, very different now. And at that time, there would be tides running through between 6 and 8 knots, which we certainly don't see anything like that nowadays. In Scapa Flow Pass, this is a kind of a graphical reconstruction, just putting these arrows onto the hydrographic charts to show what the old Scapa would have been like in flood and what it would have been like in ebb. And I think you can see at the top there, these arrows coming right across and back the other way. Obviously, with the addition of the block ships, and I really like this aerial photo because it really does illustrate, the power of the tide running through there. And you can see very clearly the wake behind the block ships there, and imagine the power of that running through. And try to imagine in your mind how that would be for the marine life and the organisms living on the seabed there, compared to if you don't get that strength of flow moving through. So this is a diagram of the Scapa Flow defences between 1940 and 41, and you can see there's quite a lot of different structures there because there's the boom defences, there's guard and mine loops, indicator loops, anti-boat nets, and all manner of different things. There was quite a lot of obstructions and things going on at that time. And then eventually the barriers themselves got made much more permanent by the addition of the concrete and glass mixed blocks that were put into place, and that really formed the basis of what we see now today at barriers 1 to 4. And obviously you can see this much more clearly from this aerial photograph, just how significant these barriers are. And so just to kind of really reinforce that, compared to the previous diagram I showed you, you can see now modern Scapa emptying and modern Scapa filling. And really this contribution that we would have had from this side is really just not there nowadays. And interestingly, when I've dived in those areas closed into the barriers, I do see remnants of horse mussels, for example, populations that perhaps would have been very dominant in times gone by, but now we just see occasional individual horse mussels, very large ones, and I wonder whether they are the remnants of what would have been previously much larger horse mussel beds in that area. So I'll come on a little bit more now into the marine life, having explained these sort of major physical barriers that have affected the conditions. And so obviously the other big thing that happened was the scuttling of the German fleet. And what that did was actually provide a hard surface substrate in areas on the seabed where previously that wouldn't have been available. So these are like islands of hard substrate which then marine life could come in and colonise. And then there's also the thinking about what was actually there on the seabed before the wrecks went down and impacts on that. So many divers, as you will have heard, come to Scapa Flow because they are absolutely fascinated by the story of the German wrecks. And they really want to come and swim around and see these and experience these massive structures for themselves. From my own personal point of view, I think wrecks are fascinating, but I'm really much more, as a marine biologist, looking into the marine life and interested in that side of things. So my experiences in Scapa Flow have been much more based on surveying the marine life, and initially I was doing that through a citizen science programme, which is called Sea Search, and some of you in the room may have heard about that, or may have even been involved in it yourselves. So in 2011, I was involved in a survey up at Fair Isle, which started in Scapa Flow, went up to Fair Isle, and then came back into the flow at the end of the week. And our last dive in Scapa Flow was on the SMS Karlsruhe. You know, many of the divers that will come to Scapa Flow will be looking at the rusty bits and the shiny bits on the wrecks. And I look at a lot of these structures. Sometimes I know what they are, sometimes I don't. But I'm usually more looking at this kind of thing, which is the fluffy stuff, or the turf, or the squidge, as some people like to call it. And to me, you know, I get a great pleasure in understanding what's on the wrecks, why it's in those particular places, and what it's actually doing in the ecosystem. And so things like these beautiful plumose anemones were often found on the tops of wrecks or on corners where you get a really nice flow of current coming around. And that's really because they are filter feeding animals and they need the food to come to them. And they'll gather around in large groups because at the base of the stem of the anemone, they actually can reproduce asexually. So you get little pinches of tissue coming away from the base, and they'll move away and grow into a new anemone. So that's why you can quite often get large clumps of them growing together. When I get my nose down and my camera going, I can get really ensconced for hours, and many wildlife divers get very fascinated by this particular group of organisms, the sea slugs. And this is a species called Gerontomentosa, and many of the sea slugs have a preference as to what kind of thing they eat, and this one's actually grazing on this finger sponge, Haliclona, which is his particular favourite food type. And by doing this, the sea slugs are transferring energy from these sessile organisms into their body, and then other things will come along and obviously eat the sea slug. So we're starting to see a bit of a chain of energy transfer through the food web around these wrecks. And a little bit deeper down on the Karlsruhe, there's a section towards the bow where there's a lot of anchor chain lying on the seabed. And here is a great place to see the sea urchins. These are related to the starfishes. And the sea urchins are very important because what they're actually doing is performing a grazing function. And so all these surfaces will get scraped over a period of time by these large numbers of urchins. And again, what they are doing is creating a space, a new area, a clean area where other things can come along and settle. So they are basically important in that process of what we call succession in the marine environment. And when we start looking at the area just a little bit adjacent and slightly away from the Karlsruhe, we get into an area which I find particularly fascinating. And it's an area I've been to time and again on my dives in Scapa Flow. And this is an area where we get a large aggregation of these large mussels called horse mussels. And these horse mussels are really important to the marine environment because they are acting like a huge biological pump. And what they're actually doing is taking particles out from the seawater, and they're actually sucking them into the stomach, digesting and converting all that food material into hard calcium carbonate shells. And when they grow together in large groups like this, they can form quite long-lived and long-lasting structures on the seabed. And as you can see here, in amongst the horse mussels, just poking out there are many other hundreds of marine species living in amongst there. So they're very, very important for biodiversity in terms of being a hotspot. And just to show you, in Scapa Flow, we see this assemblage where we get horse mussels and brittle stars, which is another kind of starfish, living very much in a close association. And if we just move a few of the brittle stars away, you can see that we get these, in some areas, clumps, and in some areas, much more dense actual carpets of the horse mussels. And of course, wherever you get food aggregating in large amounts like this, you're also going to come across predators. This predator is the Luidia ciliaris. It's a large predatory starfish, and there is a healthy amount of these starfish, I can tell you, in Scapa Flow. And they'll move through the brittle stars and eating the brittle stars and also eating the mussels and digesting the mussels. And they can be very fast moving if anyone's ever observed these moving along. There's quite a strong reaction between the predator starfish and the prey brittle stars. If you ever watch these on a video, it's quite interesting to see how the brittle stars will move out of the way because they can sense the predators coming. And I just want to spend a couple of minutes, while I've got everyone's attention, to extol the virtues of these horse mussels, because I don't very often get a captive audience like this. What the horse mussels are these bivalve-shelled mollusks, and they do act like a pump, and they secrete this shell made from the calcium carbonate. And the shell itself made of different types of minerals called calcite and aragonite, and they're laid down in layers. And they can be very long-lived. Horse mussels can live 70 plus years, and as I said, they can form a stable habitat. And the way they do this is by spinning out threads from the body made of protein, and these threads are called byssus threads. And the byssus threads bind together, the individuals bind together, and they can be really stable, and it's almost like a bit of a biological glue, if you like. And they will trap in other bits of shells, other bits of seaweed, other bits of debris, and the whole thing creates an area where other things like small worms, hermit crabs, lots of other different small creatures can actually get in and live in among the horse mussels. So the fact that they actually can build this habitat for other organisms to live in means that they have a special conservation status as a priority marine feature. And these are the types of features that Scottish Natural Heritage have the need to monitor and protect. And I just want to recount an experience that I had on this last dive coming back from the Fair Isle trip, because we went to do our dive on the Karlsruhe, and we went down the usual route, down the shot line, over the wreck, up towards the bow, and then off onto the horse mussel reef. And I was just searching around, taking a few pictures, writing on my slate, everything that I could see, and I came across this thing. And this is another kind of bivalve mollusk, but it's a very, very rare and unusual one. And this is actually something known as a fan mussel. And it's the sort of thing what I've read about in books, but I never ever thought that I would see one in real life, because they're so rare. And they're usually living in much deeper water. So to find one not 15 metres away from the wreck of the Karlsruhe was a really, really exciting moment. And if we just zoom in a wee bit closer, you can see the bright yellow mantle tissue there, and it's open and filtering. But when you see it in real life, it's only sticking up about an inch from the seabed. If you see one that's been dug up from a museum collection, this is what the actual shell looks like, and they're far bigger and extending right down into the sediment. And they used to be a lot more dominant in our seabeds, but they've suffered really from the impacts of dredging activities. So again, it's a species that anchors into the seabed with the byssus strands, and a little bit of a folk tale. These byssus strands are so like human hair that some fishermen believed that fan shells had fed upon drowned people. And they deemed that the species was unclean and they would return them to the sea if they were caught. The shells have been very popular in the curio trade, and this is a fascinating thing that people have even made cloth out of them. And I do believe similar species in the Mediterranean, people still do weave cloth from the byssus threads of this similar species. I just wanted to delve into this a bit more detail because I wanted to find out how much data was really known about the species. Looking back into the NBN Gateway and also in the Natural History Museum in London, going through the catalogues there, fan mussel records date back from 1764 and up to the present date. 86 out of the 111 records were recorded only within the last 10 years. And 84 of those records came from a drop-down video survey conducted in the Sound of Canna in 2009. So very interesting that we found one in Scapa Flow, and I can't help feeling that there must be more. So the citizen science divers have got a job of work to do to go out and find where they are. And I want to move on to another priority marine feature, and this is something that we found at the salvage site of the SMS Moltke. And I want to show you a short video that was taken by the remote operated vehicle as part of the Ship Time project in 2017. And I was very kindly invited to join the survey by Sandra Henry, who at that time was working with the Orca unit in the Orkney College. So we spent a few days out on the Scotia vessel in Scapa Flow with a specialist team of guys operating the Predator. So we would sit in the cabin in the Scotia, basically just staring at a screen all day, and the two guys would be controlling the ROV. So you can see wreckage there, and then we're panning around, and you can start to see these funny looking patches appearing into view. So I was asking the ROV driver if there was a way of just, getting in a wee bit closer without actually getting the tether caught up on the wreckage, which is always a bit of a nerve-wracking thing when you operate these kind of survey machines. So I was starting to get a bit of an excitement when I was watching this film, because I could see some kind of structure there, and I wanted to understand better what it was. Eventually, we could get in close enough, and I was getting the sense that I was looking at these big mats of seaweed. And there's only one creature I know that uses these bits of seaweed to make its home in this way. And I was getting the distinct impression that these mats could be flame shell beds. So we used that evidence from the ROV and the marks to go back with some divers and to really try to ground truth this a little bit better. And as you can see, my hunch proved correct, and I was able to make this short film to indeed check that there were living flame shells there and that these were indeed the nest structures of the flame shells. And you can see here again, this is a very active mollusk with the bright orange tentacles, and it has a jet propelling mechanism to allow it to move around and build its nest and bind these bits of seaweed and things together. It's a very clever architect and quite powerful and able to move the structure around and mould the structure to how they want it to be. And so in amongst all this matting would be tunnels and galleries where the flame shells can move around through the mat, be hidden from predators and live safely. But at the same time, building this whole habitat, and you can imagine lots of other creatures will also benefit from that. And previous work on flame shell bed biodiversity has indicated more than 200 species can live in and among the flame shell nests. So again, another amazing biodiversity hotspot in Scapa Flow. And just a few still photos from the same site. Again, it doesn't really look that very exciting on the face of it, but these piles of seaweed getting bound together and creating these structures. And a little bit closer here, you can see the beautiful tentacles there. And these tentacles contain a toxin which puts things off from attacking the flame shells. But unfortunately, this toxin doesn't work against all creatures. As you can see here, the velvet swimming crab is quite partial to a flame shell or two, and not to mention the odd octopus, also a keen predator. So I just want to finish off by saying that, in my view, the World War I and later wrecks now act as artificial reefs in Scapa Flow. And there's a very diverse marine life living on the wrecks and in the close vicinity of the wrecks. And this is including habitats of very high conservation value that, in my view, have been allowed to thrive, particularly because the wrecks and the salvage sites are there. And these habitats are of significant interest, not only because of the biodiversity, but also because of their ability to sequester carbon. And some of you may have heard about this terminology, blue carbon. And this is the carbon that's sequestered into our marine ecosystems, particularly in the inshore environments. And in the last 12 months, we've been very fortunate to be involved in a new study to look into the role that these kind of habitats can play in the mitigation against climate change and understanding how these habitats can contribute to long-term stores of carbon. I think that the association of the wrecks with the blue carbon habitats is very unlikely to be a coincidence. I think these habitats have thrived because the wrecks are there. And I think this is very important to consider in the future of Scapa Flow. And I want to thank all these different people that I've been able to work with in the last few years since I joined ICIT to pull together this thinking and these ideas. And now we're actually starting to really get the evidence and the data sets together to show that the thinking is actually real. And with that, I'd like to pass over to my colleague, Dr. Andrew Want, who's going to tell you a bit more information about the Orkney Blue Carbon Audit project. Thank you.
Thanks, everyone. It's a real pleasure to be speaking to you today. A real privilege to be at this conference. As Jo mentioned, we're working together on a project titled the Orkney Blue Carbon Audit, and she's explained what blue carbon means. It refers to carbon that's stored in marine and ocean biological habitats. It also sometimes includes the sediments that you find on the bottom of the marine environment as well. So I want to talk to you now a little bit about the project that we've been working on in greater detail. Firstly, Joe and myself with Heriot University are leading this project, but there are several other partners I wanted to acknowledge. Aquaterra, Gareth Davies will be talking shortly, and in addition to that, they're a GIS-based team led by Duncan Clark, and the map, which will be seen later, produced by Chris Page. We also have some colleagues within academia, which include SAMS in Oban, the University of Glasgow, and the University of St Andrews. Our blue carbon audit is actually looking at all of Orkney waters, not just Scapa Flow, although Scapa Flow is obviously a very, very important component of that. We're looking at the Orkney defined by the 12 nautical mile boundary around it. And because of the interest of the importance of the Loch of Stennis, that's also included as well. The only thing that we're not including is that technically around Sewell Stack and Sewell Skerry there is technically that's Orkney as well, but for the purposes of this survey we haven't included those. So what are these types of blue carbon habitats that we're speaking about? Well, blue carbon, of course, anything organic, any biological system can be collecting carbon, but the main ones we're thinking about are things such as the very extensive kelp forest which we find, seagrass meadows. These actually have, in their sediment of seagrass, has very large amounts of carbon stored there. Salt marsh is an intertidal area. This is Walkermill Bay. So in the Scapa Flow area, the Brigger Waith is a great spot for salt marshes. Walkermill Bay is mentioned, and actually Kirkwall Bay. You think where Scapa Beach is, that whole area there, that beach is largely formed because of the road system and the concrete support there has filled in with sand, but that whole area of Kirkwall Bay, the lower area towards like Cranton and places like that, those would all be salt marsh if it wasn't for human intervention. Other habitats include merle, which is this coralline-like algae that you commonly find. That's a very, very important source of blue carbon. As Joe mentioned, horse mussel reefs are very, very important, and the flame shells we'll talk about a little bit later. Bryozoa are a type of colonial animal, which are a major contributor to carbon stocks as well. Brittle star beds, and lastly, marine sediment. So just to reiterate this again, underneath where the organisms are growing, you sometimes will get then a concentration of organic material there, of blue carbon being stored there, and that's an important area to look at as well. In fact, as we'll find out later, the marine sediment is the dominant source of carbon being stored. So what we did for our survey is a four-stage approach. Firstly, we wanted to do surveys to gather as much data as possible on the abundance of these different habitats. We prioritized the previous habitats I mentioned. There are, of course, other biological habitats out there. We don't have the ability to survey absolutely everything. We prioritize those main habitats, but there are some other important ones that we didn't have the opportunity to look at yet. So anyway, our four-stage approach. First one is to do surveys to assess the abundance and distribution of where these habitats are found. The second is to take that data, as well as all previous recorded data, in order to inform models of where we would find the organisms and where you would predict to find them. So areas which we haven't yet surveyed, we can make predictions about the likelihood of what type of habitat will be found there. The third stage is to get concrete values of how much carbon is actually in those habitats, and that is also something that is lacking in many of these environments. And lastly, to get our final value for how much carbon there is a combination of those previous three. So I need to talk to you now about the various stages involved. So stage one, in great detail. So what we used were diver surveys, so going down there and checking the abundance of the organisms and habitats. We also use ROV and other drop-down camera technologies, another way of, again, surveying what's out there. So that's our way of improving our knowledge of where these different habitats are found. The second stage is to use this predictive mapping. It's a bit complicated, and I don't want to get into great detail about it, but it's a system where using a GIS base, you inform the model with various layers of different environmental variables in order to predict the likelihood that you'll have the correct conditions for that particular type of habitat. So let's say you're looking at kelp, for example, you'll look at what the substrate is it an appropriate substrate for it, what is the depth, and all sorts of other factors to see how likely it is, and you make a prediction based upon those different factors. So these different factors include things like substrate, as I mentioned, and this absolutely includes wrecks. So the wrecks in Scapa Flow are a very important substrate for organisms which encrust and grow on hard substrates. Other things, light penetration, this is of course to do with photosynthesis largely. Wave exposure and tidal hydrodynamics are very important factors also to consider. So what you do with stage 2 then is, as I mentioned, there is this model that we work upon, and so using the example of brittle stars, we are able to combine known records with the model and then predict the likelihood of any given location where you would expect to find brittle stars. So based upon these different factors that we looked at on the previous slide, we see that in Scapa Flow, that's a prime habitat for brittle stars. And as Joe showed earlier, you sometimes get these very, very, very dense beds of brittle stars. The third stage was to get some hard data on how much carbon is found in a given habitat. So we got samples from the diver surveys, etc., and other sources, and we do a procedure called a loss on ignition, where we heat the samples up. We heat them up to 550 degrees, and that burns off organic carbon. Let me clarify, organic matter. It's a little bit of a caveat. If anyone's interested in greater detail, I'll be happy to talk to you. But basically, heat up to one level, gets rid of the organic matter, which is mostly carbon. You heat up again to 1,000 degrees, and that gets rid of the inorganic carbon. You weigh it in between at different stages, and by doing so, you find out how much carbon is found inside the organisms. To determine the overall calculation, it's a fairly straightforward calculation at the end. You know how much habitat you have, where it's all found. You've got to find out how thick it is, so third dimensionally. So is the merle 30 centimeters? Is it 90 centimeters? And that can sometimes be a bit tricky to find out. And then if you know how much carbon is found in any given amount, you then know your total carbon. So we have done this for those various different habitats, and I focused here on Scapa Flow. So we have, essentially, we have a variety of different amounts of blue carbon. This is the biological blue carbon. So this again means the blue carbon that's actually stored or sequestered into living organisms like kelp, brittle stars, horse mussels, etc. It doesn't include the sediments itself. To go back to that a second, sediments actually, it turns out, are the dominant store of carbon. Things like muds and sands and the rocks may be where you find the majority of the actual carbon stored. To give you a couple more specific examples of some of these organisms, with kelp, you don't get a huge amounts of kelp in Scapa Flow itself. I want to show this as a contrast to Scapa Flow. With kelp, you find it typically close to the shore. The reason why, of course, is because kelp is photosynthetic. So if you get further to deep waters, kelp cannot thrive because it's too deep. And it especially likes the high wave exposed shores of West Mainland. Once you get into Scapa Flow, the amounts of kelp significantly drop, but it's still obviously an important resource Orkney-wide. In contrast, as Joe was highlighting at the end of her talk, the flame shells, Limeria, these are found in, it's a perfect environment for them within the waters of Scapa Flow, especially the areas around Carver, Farrer, and Rise a Little. So there are some important German wrecks in these waters, and again, based upon the records we have and the predictive modeling, this is the area where you find the largest, or expect to find the largest amounts of the flame shells. This area is also of interest for the scuttling, is where a lot of the breaking up of the wrecks occurred. So that area also has a lot of wreckage that remains there, again, close to Rise a Little, etc. And this may be a very important area that allows the flame shells to still survive. And another reason why they're found particularly in this area is because they like this tidal flows that you get between the narrow channels between the islands. So I just want to move on and kind of wrap things up with what are the challenges to the study that we've done. It's a very important study we've just completed, but it is a step in the process of trying to understand better how much carbon our marine habitats store and how we might be able to manage that more effectively. So we're trying to always trying to get more data, always trying to understand the distribution better. These models are only as good as the data that feeds into them, so the more information we get on distribution, the better the models will become. Further ground truthing is necessary to validate these models. I also mentioned that you have to understand it three-dimensionally as well. It's well and good to understand how broad an area of Merle is, but you also need to understand it from the third dimension as well to know the total amount of Merle. And the depth of a lot of these sediments and a lot of these habitats is very poorly understood. There's huge variation in Merle. Traditionally is thought of being fairly superficially on the surface, but we've been doing work in Wyre and Rousey Sound where we've found Merle extending well over a meter in depth. Preliminary results of the carbon dating of this suggest that it is several thousands of years old that it's been accumulating in these habitats. And lastly, an important area here is to look at the age of the stocks and to understand better the idea of sequestration. So it's all well and good to understand how much carbon we have, but we also want to understand the flux of carbon into the system and out of the system in order to hopefully guide that, perhaps possibly, towards promoting the sequestration of carbon to lock it into marine habitats as a means of mitigating against climatic change. So a lot of things are mysteries that still remain unknown. We're all familiar with these huge wash-ups of kelp, the tangles on the shoreline. Where exactly does all this kelp go to? We don't actually know for sure where in the cycle of carbon this goes to. Are there sediment layers that are just packed full of kelp originating carbon? We don't properly understand. So I just want to finish with what the main achievements so far of this audit are. We have been able to enhance and improve our knowledge of the distribution of these different habitats throughout Orkney. We have improved our understanding of specific amounts of carbon by doing tests ourselves on samples of brittle stars and the sediments and the salt marshes. Overall, the whole story has been improved and we are learning more and more all the time. It's also provided a wonderful opportunity to work with a whole bunch of different organizations. We've had input locally from citizen science, we've had input from aquaculture, we've had a lot of support from the regulators like SNH, for example. It's been a very positive experience. What the main output of this is going to be will be a report. I already gave an acknowledgement to the people who worked on the project. I just want to give an acknowledgement to the people who have also supported the project in various ways. There's a whole list of people, including our funders, and including other people who have been supplying critical data for the survey itself. That's it for our presentation on the Orkney Blue Carbon audit. Thank you very much for listening.